Hi everyone, welcome back. This week we're going to be finishing off our second part of Chapter 6, where we look at non-Mendelian inheritance patterns as well as genetic disorders. So if you think back to last week, we had focused primarily on Mendel and his work. We were looking at a field of genetics that looks at complete dominance, right? If we had two different alleles, one was always going to be dominant over the other. And if we blend those two together to create a heterozygous individual, the dominant allele would always be expressed. So this week we're moving into non-Mendelian inheritance patterns, which will follow a set of different rules. So the examples we're going to be looking at are polygenic inheritance, incomplete dominance, codominance, such as blood typing, epistasis, environmental factors, as well as X-linked inheritance patterns, where we take a look at how alleles located on the X chromosome can be passed on to both female and male offspring. Now, last week when we looked at, complete dominance of one allele over another, we also had to make some assumptions. We had to assume that the trait that we were looking at, that it would only be coded for on one specific chromosome. And when we looked at multiple traits, we also had to assume that the genes coding for those traits were going to be located on separate chromosomes. And so this week, the focus really is then going to be on all of those circumstances under which those assumptions cannot be made. So one example that we talked about last week was height, right? It's not that we have tall and short. We have a wide variety of heights distributed within our population. And this is because height is showing us continuous variation, where you don't just have one dominant over one recessive. You have a trait that's showing a range of differences because you have multiple genes acting together that are ultimately going to be determining that trait. And here you can see the distribution of individuals in the photo. This here is your ideal bell curve, where you have the median in the population as well as the average in the population meeting up perfectly here in the middle. This doesn't always hold true, but you can see here we still have a relative normal distribution with heights within a population. The point to make here is that heights, we don't just have tall versus short. We have a whole range of different expressions. And so height is just one example of a trait that displays this continuous variation within a population. Okay, so not only height, some other examples, weight, skin color, and then also a number of diseases. Right? Remember, polygenic inheritance is that where there isn't just a black and white, there isn't a dominant and a recessive. You have multiple factors, multiple genes that are playing a role into the expression of that trait. And as a result, the phenotypes that you see vary significantly. Now, the second non-Mendelian inheritance pattern we're going to take a look at is incomplete dominance. Okay, so last week we looked at complete dominance, where you had two alleles, a dominant allele and a recessive allele. Now, with incomplete dominance, you have alleles that are not fully dominant or fully recessive in their heterozygotes. With incomplete dominance, the alleles are equally expressed, and so as a result, there's an intermediate that is formed by the heterozygous genotype. Remember before, if we had a dominant allele that was red and the recessive allele was white, if we had a heterozygous individual in complete dominance, the heterozygous individual would display that dominant phenotype. Okay, but with cases of incomplete dominance, the alleles are neither dominant nor recessive, and so they are blended when we see the heterozygous with one of each allele. And so in order for us to keep this straight, we use a different type of nomenclature to be able to describe and identify the genotypes of the incomplete dominant individuals. And you'll notice that the way that we write this is instead of just having the allele recorded, we have the identifying allele written as a superscript to a capital C. Okay, so seeing the capital C here indicates to us that we are looking at an individual that is displaying incomplete dominance. And so here's an example. If we take your parent generation, you have a homozygous red parent and a homozygous white parent. Notice, because one allele is not dominant over the other, we are going to be writing this differently. Okay, so the red allele, you give it an R, the white allele, we're giving it a W, but we're using capitals for everything because this is showing us incomplete dominance. So the way we describe this is that we have a homozygous red parent and a homozygous white parent. Now, if we blend those two together and you complete a Punnett square, you're going to see that 100% of the offspring from this F1 generation from two homozygous parents are going to produce an intermediate phenotype. And so when you combine a red allele and a white allele, we create an intermediate genotype, which is C to the R and C to the W. And the phenotype of that individual is pink. Now, if we take that intermediate heterozygous individual from the F1 generation and we self-cross them, we're in a situation where we're going to see the genotype ratios that existed before. You're going to have one homozygous red to two heterozygous to one homozygous white. And now the difference here is that we're also going to see a 1 to 2 to 1 ratio, because remember, the heterozygous genotype, C to the R, C to the W, that is going to produce the intermediate phenotype, which is pink in color. And so the genotypes are 1 to 2 to 1, and the phenotype ratio for incomplete dominance, 1 to 2 to 1, when you cross two heterozygous individuals. Now, the third type of non-Mendelian inheritance pattern is codominance. And with codominance, there's not necessarily going to be a dominant allele again, but instead both alleles can be expressed. And so the example of codominance we're going to be taking a look at today is our blood type. Now, it's important to mention there are two different systems used to identify an individual's blood type. First, there's the ABO blood type system, right? And that's what gives you either your type A, your type B, type AB, or type O. The other system you can see identified here by whether you are positive or negative. And that positive or negative is an indication of whether you have a specific type of antigen known as the Rh antigen on the surface of your red blood cells. So if you do have the Rh antigen, you are positive type of blood. If you don't have the Rh antigen, you are negative. And this plays a role just as equally as to whether or not you're going to be compatible to receive a certain type of blood. Our main focus, though, for today in terms of codominance and inheritance patterns is going to be to look at the ABO system. Now, there are specific genes that determine your ABO blood type, and they exhibit more than one dominant allele. There's really only two types of antigens, which are the recognition markers on the surface of your red blood cells. We have A antigens and we have B antigens. We have either A antigens or B antigens. And as you'll see here, if you have A antigens on the surface of your red blood cell, you're type A. If you have B antigens, you're type B. Now, if you're type AB, it's because you have both A and B antigens on the surface of your red blood cell. And if you're type O, it's because you do not have any antigens on the surface of your blood cells. And now the genes that are encoding for those enzymes that are going to be creating those antigens for your red blood cells, they have three different alleles. So we have uppercase I to the A, uppercase I to the B, and your lowercase I. These are your three main alleles in your codominant case of blood typing. The combination of two of these three alleles will dictate whether or not you are type A, type B, type AB, or type O. Now, the reason that we're identifying the allele where the A antigen will be produced and the B allele where the B antigen will be produced with a capital is because these two alleles are dominant over this third allele. And this allele here, the lowercase I, codes for no antigens on the surface of your red blood cell. So remember, it's going to be a combination of these three. So you can have two of these. You can have two of these. You could have two of these. Or you could have combinations of these. So you could have an A and a B. You could have an A with a lowercase I, or you could have a B with a lowercase I. All right, and this is what together is going to add up to creating our four different blood types. Now, not only are A and B dominant over the lowercase I allele, but they are also codominant. And so if you have both the A and the B allele, you are going to produce both A and B antigens. And so here's an example when we take an allele from either parent of the 6 possible genotypes that we could create. So as I mentioned before, the A antigen allele is dominant over the lowercase I allele, which codes for no antigens. And similarly, the allele that codes for the B antigen is dominant over the lowercase I that codes for no antigen. So when we combine these alleles together, we get our genotypes, the 6 genotypes. Let's follow through. If you have 2 copies of the A allele, your blood will be type A. If you have 1 copy of the A allele and a copy of the no antigen allele, you will also be type A. However, you're a carrier. If you have 2 copies of the B antigen, you will be type B blood. 1 copy of B, 1 copy of lowercase I, the no antigen allele, you will also be type B blood. Now remember, A and B are also co-dominant, so we have another type. If you have both an A and a B allele, you will be type AB blood. And now this is where it comes into play as to whether you're a carrier or not, because you could have parents that are type A and type B, and the offspring could have type O blood. All right, and so with type O, remember type O blood, we don't have any antigens on the surface of our red blood cells. And remember, both A and B are dominant over the lowercase I. So you have to have 2 copies of this third allele, which codes for no antigen to be produced, in order for yourself to be type O. All right, so it's important first off that you understand what the genotype is coding for in terms of a phenotype. We have 4 different phenotypes: type A blood, type B blood, type AB, and type O. We have 6 different genotype combinations that could possibly yield these 4 phenotypes. So make sure you remember there is codominance, so A and B can both be expressed in type AB, and then your A and your B allele are both going to be dominant over your lowercase I. Now, when determining who can donate blood to who, we have to consider what type of antigen do you have on the surface of your red blood cell. If you are type A, then your body is going to recognize the A antigen that you have on all of your red blood cells as a normal antigen. It'll recognize it as self. What your immune system is going to do, though, is produce anti-B antibodies. As you can see, the antibodies will bind perfectly to the B antigen because it recognizes the B antigen as something that's foreign and that shouldn't be in your body. And so if you received type B blood or type AB blood, if you're a type A individual, then your anti-B antibodies are going to attack those cells and clump them all together, rendering them useless. All right, so this is why you have to make sure you know what a person's blood type is before giving them a blood transfusion. So if you're type A blood, you have the A antigen, you're going to make the anti-B antibodies. Now, if you're type B blood, you're going to be making antibodies against A because you're not going to recognize A antigens as self. You'll recognize them as a foreign antigen. So type B blood individuals, they produce anti-A antibodies. If you're type AB, you have the presence of both A antigens and B antigens on all of your red blood cells. And so your body is going to see both of those antigens as normal. That's why individuals with type AB blood, they're known as the universal recipients. Because they recognize both the A and the B antigen, their immune systems don't produce any antibodies against any blood type. And so they can receive B because they recognize it as self. They can receive A because they recognize it as self. And they can obviously receive O because O doesn't have any antigens that would help our bodies to detect anything on it as a foreign blood cell. And that brings us to our last blood type, type O. Now remember, for type O blood, you need to receive both the lowercase I alleles in order for no antigens to be expressed on the surface of your red blood cells. Now, because you do not have any antigens, well, your immune system is going to produce antibodies against both. And so type O individuals can only receive type O blood. If they receive any of the other 3 types, their immune system is going to attack those blood cells because it doesn't recognize any of those antigens as self. Individuals with type O blood are known as universal donors. And that's because they don't have any antigens on their red blood cells. And so you can give type O blood to an AB individual and, well, they don't have antibodies anyway, so they're not going to attack the type O blood. You can give type O to a type B. Type O doesn't have any A antigens for the anti-A antibodies to bind to. And then similarly, type O can be given to type A. because there are no antigens and therefore their anti-B antibodies won't be able to attach or attack anything there on the surface of the O blood cell. Okay, so type AB are known as the universal recipients and type O known as the universal donors. Now it gets a little bit more complicated if we go back to looking at both positive and negative, whether you have the Rh antigen or you don't, but for the purposes of our lecture today, we're going to be just focusing on the ABO blood typing system. Now, this Punnett square here is a 3 by 3 because we have 3 different alleles in different combinations that could be given from one parent to the other. What I need to make sure you understand is this is showing you in blood typing every possible ABO blood type that could be created from those 3 possible alleles. But remember, each parent is only going to possess 2 of these 3 alleles. So if I gave you an example and said the father is type O and the mother is type AB, well, there you have your 2 genotypes. Now you can create your 2 by 2 Punnett square, and you can cross those alleles to figure out the likelihood of producing offspring with a certain blood type. Now, just taking a look at this, what do you think the chances are that from 2 parents, one that is type O and another that is type AB, what do you think the likelihood is of having an offspring with type O blood? Well, right away, hopefully you're looking at this thinking, well, it would actually be 0 because it doesn't matter how you work this out, you need another parent to be a carrier in order for there to be a possibility of an offspring having type O blood. Okay, and now we're going to look at epistasis. Now, epistasis refers to the fact that you can have one gene that is dependent on the presence of one or more modifier genes. In this example, we're going to look at fur color of Labradors. Now, there are 2 different genes at play here. And so you may be looking at this originally thinking, okay, this looks a little bit like a dihybrid cross. You have a 4 by 4 square. Well, think back to Mendel, right? With Mendel, we were looking at traits that were independent of one another. In this example of epistasis, the modifier gene, identified by the letter E, dictates whether or not the B genotype for fur color is going to be expressed. So we're looking at 2 genotypes here. We have the genotype for fur pigment, and we have the other genotype, which is the modifier. And the alleles for fur pigment are either uppercase B or lowercase b. And so we have homozygous dominant, heterozygous, and homozygous recessive. As you can see here, homozygous dominant, heterozygous, and then other examples here, you can see homozygous recessive. The dominant allele is completely dominant over the recessive allele. Okay, and so your homozygous dominant will look black. Your heterozygous will look black. The homozygous recessive will look brown. Now that we've established that, you then need to take a look at the modifier gene. If the modifier is in its homozygous recessive state, it will then shut off the other genotype for fur color. And so anytime you see a homozygous recessive modifier gene, that is going to shut off the production of fur pigment. And so all of those dogs end up looking like yellow labs. So start off, whenever you're looking at interpreting these, your homozygous dominant and heterozygous are all going to express the dominant allele. Your homozygous recessive will express the recessive allele, which in this case is less fur pigment. Okay, so they look brown. And in all of these cases, you can see here the modifier is either in its homozygous dominant or heterozygous state. Okay, look through all of the black and all of the brown. And the only time we end up with a yellow lab is when the modifier gene is in its homozygous recessive form. Okay, what you do need to understand is that with epistasis, one of the 2 genotypes will be the modifier and one will be the gene in question. Okay, so for this example here with fur color, if you did not have the presence of the modifier gene, you would have black, which would be the dominant color, and brown, which would be the recessive. Okay, so homozygous dominant and heterozygous would look black, and then homozygous recessive would look chocolate brown. Because we have the addition of this modifier gene, which can shut off our genotype, when the modifier gene is in its homozygous recessive state, it will shut off the gene for fur pigment, and as a result, you'll end up with a yellow lab. Now, as I mentioned before, we can have one or more modifier genes. So you can see this could get very complicated very quickly. So for simplicity's sake, we're sticking here with the one type of modifier gene. And in that case, it needs to be in its homozygous recessive for that modifier gene to override the gene for fur pigment. Now, environmental factors can also play a huge role in determining what type of alleles will be expressed under certain environmental conditions. And here we're looking at two examples here of Arctic foxes as well as the flowers of hydrangeas. Now, with the Arctic foxes, they have heat-sensitive alleles, and these alleles are turned on and off by temperature. And so the only time that the alleles for fur color are going to be producing pigment is when the temperatures are warm. And then when the temperatures are cold again, the alleles that are going to be producing that fur pigment are going to be turned off. And so the Arctic fox then does not have any pigment and is able to blend in with its environmental surroundings. And you can see here how this adaptation would play such a huge role in the survival of this species. We're going to be taking a look at evolution next week, and we're going to revisit this concept here at how sometimes chance mutations can occur that can actually set up an individual to be more successful and more fit for the environment in which they live. In this case here with these heat sensitive alleles, they're capable of being able to turn on and off to be able to help the Arctic fox camouflage into their surrounding environment, and thereby making them more successful in being able to breed and produce offspring to pass on those alleles to future generations. Now, hydrangeas show differences in soil acidity. And so if the soil is acidic, the hydrangeas will tend to so if the soil is more acidic, the hydrangeas will tend to produce flowers that are more blue in color. And if the soil is more alkaline, they'll tend to produce flowers that are pink in color. And so these alleles that are determining color pigment in those flowers are pH sensitive alleles. Okay, and there's a number of other different examples. For our purposes here, we're looking at heat sensitive alleles, pH sensitive alleles. And I really just want you to take away from this that there are environmental factors such as pH or temperature that can play a role in determining how specific alleles are going to be expressed. Now, last of our non-Mendelian inheritance patterns, we're going to be taking a look at specific traits that are X-linked. That means that their alleles exist on the X chromosome. And so for a female, the female would contain two X chromosomes, XX. And for a male, they would have one X chromosome and one Y chromosome. Now, it mentions here that in fruit flies, sex is determined by the number of copies of the X chromosome. Now, that is not exclusive for fruit flies, right? It's the same in humans. And the sex chromosomes are homologous chromosomal pair 23. So if you have two X chromosomes, be female, and an X and a Y chromosome would be male. Okay, so two X chromosomes is female, and an X and a Y chromosome is male. Okay, so we're going to be taking a look here at fruit fly eye color. All right, typically you see fruit flies, they have red eyes. You can have fruit flies with white eyes, and that tends to be seen more commonly in males than it does in females. And that is because with X-linked inheritance patterns, females need two copies of the recessive allele in order for it to be expressed, whereas males only need a single copy in order for that trait to be expressed. Because remember, males only possess one X chromosome, the other is a Y chromosome, and females possess two X chromosomes. And for a female to have white eyes, they would need to have two X chromosomes each with the recessive white allele on both. So we're looking here at an example where you had a female and a male both with red eyes. But you can see here the female is a carrier for the recessive white allele on her second X chromosome. So we perform a Punnett square as we have before. You split up those alleles, right? And then we have our four possible genotypes. So here we have a homozygous red. We have a heterozygous, which will display the red in a female. And here we have males. Okay, so now with the males, it's different, right? Because we're looking at one X chromosome. So you either have it or you don't. So with this male, he has the red allele, and therefore his eyes will be red. But with this male here, he has the white allele, therefore that trait will be expressed. For a female to express a recessive X-linked trait, they need to possess two copies on each of their, they need to possess a copy on each of their X chromosomes in order for them to express that recessive trait. For males, they only need to possess one copy of the recessive allele in order for that trait to be expressed. Okay, we have three conditions here, all linked to the X chromosome. Those would be color blindness, hemophilia, as well as Duchenne muscular dystrophy. And all three of these are more common in men. And ask yourself why? Because men only need one copy of the recessive for that trait to be expressed, whereas females require two. And so in that case, think about it. You would have to have a male that contained the gene as well as a female that was at least a carrier for them to have an offspring that was both female and expressing that specific either condition or trait. Now, as I mentioned before, one thing when we were looking at Mendelian inheritance that we had to ignore was that some genes that might be located closer together on a chromosome, they may be more likely to pass on and segregate together when the parent is producing their gametes. So we had to assume for dihybrid crosses that we were always looking at genes that were located far away from each other, specifically on two separate chromosomes. The closer that those two genes are together, the more likely that they are going to be paired with one another and be passed on with one another. There's certain recessive traits as well as conditions that are more likely to occur together because they are actually located closer together on specific homologous chromosomes. First, we're going to start to take a look at genetic disorders that arise from non-disjunction. So remember, when we are producing gametes, the germ cells are going through meiosis 1 and meiosis 2 to ultimately form those 4 haploid gametes. And what we expect is that following metaphase, when all of those chromosomes line up at the equatorial plate, that then when they're pulled apart to opposite poles during anaphase, that they would separate equally. But non-disjunction can occur where there's an error during anaphase, and the chromosomes themselves don't split equally into their following daughter cells. So here we're looking at an example where during anaphase 1 of meiosis 1, we've had homologous pairs pass into the same cell. And normally we would be expecting that one pair of the sister chromatids would have passed into that other cell. And so then during meiosis 2, we now have cells that have more chromosomes and those that are missing chromosomes. And so what this looks like, by the time we've divided this diploid cell into two haploid gametes, each of them, we're going to end up with two gametes that have one extra chromosome, and we're going to have two of those gametes are going to be missing a chromosome. So depending on which chromosome was originally supposed to move into this cell and moved into this cell, we're going to end up with one extra in each of the gametes. And say this is a sperm cell, by the time that sperm fertilizes an egg, that individual that is produced, if viable, will have an extra chromosome. Now, some non-disjunctional disorders that occur, they happen, and the actual fertilized zygote that's formed isn't viable. And so if implantation were to occur, the pregnancy would be terminated with an early miscarriage soon after implantation. And what can happen is when this happens in sperm cells, there's so many sperm cells produced, those with extra or missing chromosomes may not be viable in and of themselves, and so wouldn't even be able to fertilize the egg. Now, the term that we can use to describe whether you have additional chromosomes or whether you have missing chromosomes is aneuploidy. And with certain types of non-disjunction, depending on how many chromosomes are missing or how many extra chromosomes you have, the gamete itself may not be viable, or even the fetus that's produced may not be able to develop to full term. Now, certain non-disjunctional disorders, such as trisomy 21, individuals with trisomy 21, where they have an additional 21st chromosome, they're able to live well into adulthood and to thrive. Now, individuals with trisomy 21, which is also known as Down syndrome, they typically experience developmental delays or even mental impairments. So a lot of the non-disjunctions, the errors that can occur during cell division and the production of the gametes, sometimes the gamete itself is not viable. Remember, in a female, only one of the 4 haploid gametes that are formed will actually develop into a mature egg cell. Now, we were talking about non-disjunction of the 21st chromosome. In certain circumstances, you can also have non-disjunction of the 22nd chromosome, and so trisomy 22. Non-disjunction can also happen with the sex chromosomes. Okay, so remember, your sex chromosomes are number 23. We have 22 that are autosomal, and then the 23rd pair are the sex chromosomes that determine your gender. So when the gametes are being produced, ultimately the male is either going to be giving an X or a Y. And with the female, it's always going to be an X sex chromosome that will be passed on. Now, non-disjunction with the X, where you end up with a trisomy of X chromosomes, 2 from the mother and 1 from the father, end up with trisomy X. And now the phenotype that is expressed from that genotype, female with tall stature, learning disabilities, and into adulthood, fertility issues may persist. XO, which is known as Turner syndrome, where there is only a single X chromosome. Okay, so this could be received from either the father or the mother, but the opposite gamete did not have a sex chromosome. And so a single X chromosome is known as Turner's syndrome. The individual would be female. However, because of the lack of the other X chromosome, some of these secondary sex characteristics, as well as primary sex organs, don't necessarily mature as they normally would. And so a female with Turner syndrome may be infertile as she develops into adulthood. 2 others here, Klinefelter syndrome, where 2 X chromosomes as well as 1 Y, okay, an additional X on top of the typical male genotype. Individuals with Klinefelters, we're going to take a look at in the next slide. Now, with Klinefelters, remember, this individual has an additional X chromosome. And so what you'll tend to see is that the secondary female characteristics will start to be expressed more than they would with a male with an XY genotype. So with Klinefelter syndrome, those secondary sexual characteristics that would be male would tend to be reduced, and you would see expression of female secondary sex characteristics that would start to become expressed more. So as a result, the individual typically would have smaller gonads, okay, so smaller testes, and so would tend to have issues with infertility. Now, some of the secondary sexual characteristics would be wider hips, as well as female type pubic hair patterns, narrower shoulders, as well as gynecomastia, which is the presence of breast tissue in males. Now, in addition, tall stature, lower IQ, and poor muscle tone, also some symptoms that can be seen with Klinefelter syndrome. Now, just to review, we talked about this at the beginning of the course, but a sign is something that you can measure, a symptom is something that is subjective, and a syndrome is a collection of signs and symptoms characteristically seen together as part of a specific diagnosis. All right, so with Klinefelter syndrome, you're seeing a number of these different signs that would be typical of a male with the additional X chromosome. When we're talking about genetic disorders that can be passed on through generations to your offspring, those mutations have to occur within the germ cells. Mutations that occur within our somatic cells, right? All the other cells of our body besides those that produce the gametes, those mutations are isolated to ourself. They are not necessarily passed on to future generations. For a genetic disorder to be passed on, the mutation must occur within the DNA that makes up the chromosomes within those initial germ cells. Okay, so where are they located? Within the testes as well as the ovaries. Now, those mutations that don't cause death almost always result in recessive alleles. Right? They're not usually expressed within the population as a majority because you need to have 2 copies of that recessive allele to be an individual that expresses the condition. This is where genetic screening comes into play, where people are wanting to know, what am I a carrier for? And because that comes into play, and that sometimes comes into decisions that need to be made for individuals focusing on family planning. So we're going to take a look at both dominant as well as recessive autosomal and sex-linked genetic disorders. And I've provided you here with a sample chart that you could create for yourself to help organize the information. So you have a number of diseases that we're going to be going through. You need to know whether they are dominant or recessive, and whether they are autosomal, affecting chromosomes 1 through 22, or whether they are linked to the X chromosome. So, sickle cell anemia is a disorder where the red blood cell itself does not form this typical biconcave shape. A red blood cell does not contain any nucleus or membrane-bound organelles, and that's great because we need our red blood cells to contain as much hemoglobin as possible. Hemoglobin is a protein that is responsible for transporting oxygen from the lungs to our tissues and carbon dioxide from our tissues back to the lungs. Now, we've talked about capillaries and how small and narrow they are. These red blood cells are shaped perfectly to be able to fit through those smallest capillaries. They're relatively tough, but they're also flexible in this biconcave shape. And when they're going through the capillaries, it's almost a 1 by 1 single file. Now, sickle cell anemia is caused by an autosomal recessive mutation where the gene that forms hemoglobin has been mutated, and this causes abnormal red blood cell formation. And so ultimately here, their red blood cells form what's known as a sickle shape. And so as a result, there's not only reduced capacity to carry oxygen, but because of this sickle shape, they tend to have very poor circulation as well. Now, it being an autosomal recessive, meaning that the gene is located outside of that 23rd sex chromosome and is recessive in nature, and so you need two copies in order for it to be expressed. Tay-Sachs disease is an autosomal recessive disease. There's a specific gene that codes for lysosomal enzymes. And the hexa gene that codes for those lysosomal enzymes is mutated. And with Tay-Sachs, this is something that typically can become evident in an otherwise normally developing healthy child. I've read about cases where all of the cognitive gains and development that had been occurring up to that point, the parents started to notice cognitive decline. And now that hexa gene coding for those enzymes is going to ultimately cause, at the gross anatomical scale, brain deterioration. And as the nervous tissue is deteriorating, the brain continues to atrophy and ultimately leads to death, typically before adolescence. No disease is good, but Tay-Sachs in and of itself tends to be very tragic because, you have an otherwise healthy child that all of a sudden starts to show these signs of decline and then passes before they reach puberty. With cystic fibrosis, again, we're looking at autosomal recessive. So with all three of these conditions. Now with cystic fibrosis, it's the genes responsible for producing chloride channels that are mutated. And so these individuals don't possess chloride channels. And so chlorine is unable to move from extracellular to intracellular environments readily. And so this throws off the water balance inside of the cells. And so as a result, the cells then produce excessive amounts of mucus, which can impact lots of different organs scattered throughout the body. One of the most commonly affected organs are the lungs, but also can impact liver, pancreas, as well as sweat glands. Okay, because this is a systemic issue where chloride channels in every tissue throughout the body, all tissue types are going to be impacted as the individual is unable to maintain water balance between their intracellular and extracellular environment. Now we're moving from our autosomal recessive diseases to looking at our only example of an autosomal dominant disease. And what happens with Huntington's disease is that there are repeated triplets of nucleotides: cytosine, adenine, guanine, CAG. And these repeated nucleotide sequences are inserted into the gene that increases how much glutamine. Okay, when we look at how these triplet nucleotide sequences known as codons code for amino acids later on, you're going to see that CAG in that sequence code for the amino acid glutamine. Remember, when we take the mRNA from the gene, we take that template. The whole point is for the ribosomes to read that template and produce the proteins. So what happens here is that we have a number of glutamine amino acids added into the length of that polypeptide chain. And as a result, you'll have a protein that is non-functional. This ultimately involves brain deterioration. As you can see here, atrophy of our cerebrum and also dilation there of the ventricles, those chambers within the brain. So we're going to finish off looking at two recessive X-linked genetic disorders here. First one being hemophilia. Remember, with recessive X-linked disorders, these are most commonly seen in males. And the reason being, males only possess one X chromosome. So if the mother was a carrier, then the male offspring would have a high likelihood of possessing the trait. And so hemophilia is one of those examples of a recessive X-linked disease. And this affects the gene involved in blood clotting. Specifically, there are 13 different clotting factors, 13 different proteins that all need to be activated one after another in order for the blood clotting cascade to occur properly. Now, the gene that's involved here affects the production of protein number 8. Okay, so it just affects one simple blood clotting protein, but that interrupts the entire cascade of the blood clotting process. All right. Now, lack of any of the blood clotting factors can ultimately lead to an inability to stop bleeding. So, hemophilia, typically, though, it's clotting factor 8 that is impacted. And as a result, they are unable to activate the blood clotting process and stop bleeding. So here's an example where we're looking at inheritance of hemophilia. In order for the offspring to have hemophilia, the mother needs to at least be a carrier. Because remember, if the mother's not a carrier, well, the males are only going to get the Y chromosome from the father, right? And then they won't get anything from the mother. And so in that case, if the mother's not a carrier and the father has hemophilia even, the children will not have hemophilia. The sons will be without hemophilia, but the daughters will be carriers. Remember, for a female offspring to be produced, the father's X and the mother's X will be combined to form that offspring. And so if the father has hemophilia, that means that the X they're going to be giving to any of their daughters is going to be carried on. And so the daughters will all end up, in this case, becoming carriers. If you looked at then that daughter who is a carrier having her own children with a father who did not possess the gene, you would see that there is a chance that her offspring that are male could have hemophilia. And that's in a way where you can see certain X-linked disorders jumping generations. Because for a female to express the X-linked disorder, she requires 2 copies of that recessive allele. Now, we're going to finish off here looking at Duchenne muscular dystrophy. You can see an image here of an individual with Duchenne muscular dystrophy. Now, with Duchenne muscular dystrophy, it is a recessive X-linked disease involving a very important protein, dystrophin, that holds the myofibrils together. And we've looked at how those myofibrils are those cylindrical arrangements of your thin and thick filaments. Dystrophin is a protein that holds those myofibrils together. Now, without proper functioning of dystrophin, then muscles will degenerate. Now, this disease affects children early on in development because the muscles end up atrophying and the child doesn't have adequate muscle tone to be able to develop adequately to move around. And Gower's maneuver sign is something that is typically looked for in developing children. Here you can see what's happening is this boy is having to leverage his own body to be able to get himself to stand up from a seated position. Typically, you would just see a child be able to use the muscles in their hamstrings, their glutes, to be able to push their body upright. And so in order for this boy to get up, what he's having to do is go into sort of a plank position, walk his toes and his hands closer together, and then ultimately push himself up from this position to be able to stand up. Okay, so that brings us to the end of our lecture for today. I recommend with the genetic disorders that you create this chart for yourself. I've given you some examples here of the ones that we looked at. We also took a look at non-dysjunctional disorders, so make sure you understand the difference between both of those. Know with the genetic disorders whether they are dominant recessive, autosomal, meaning on chromosomes 1 through 22, or whether they are X-linked. And also understanding the signs and symptoms that can result. And sometimes you might find it easier for yourself if you understand, well, what is the mutation that has occurred? And that may help you remember specifically from a signs and symptoms perspective what you could see in that individual. Okay, and that brings us to the end of our lecture for today. So if you have any questions, please reach out to me by email and I will get back to you as soon as possible. Okay, everyone, have a great week.